Bugs. They're little guys. They're a critical part of the ecosystem. They mostly belong to phylum Arthropoda. They're on your face. There are bugs on your face. They're called face mites and they live on your face. Okay, now that we have that out of the way, let's get into the episode. I'm here today to talk about bugs. Bug is a general term used on Earth to describe small terrestrial invertebrates that creep, crawl, and skitter about. Some form of bug is able to be found on almost all of Earth's ecosystems. This is because they are very adaptable and can play critical roles in food webs, acting as nutrient recyclers or food for larger organisms. Before we really get into the bugs found on Primin B though, I need to clarify something. When it comes to describing the flora on Primin B, I leave a lot out about reproduction. But after taking 2 biology classes at university, I can give some more insights on where baby plants come from. On Earth, plants reproduce with alternating generations, similar to jellyfish or xenomorphs from Alien. They have a haploid gametophyte generation that creates a diploid sporophyte generation, which goes on to create another gametophyte generation. In flowering plants, the gametophyte stage is just the pollen grain and ovule, which hold the gametes. The plant life on Primin B is going to have a somewhat altered version of this idea. In the past, I've described these little black balls as seeds and spores interchangeably. Going forward, these are going to be more like spores. We will call these structures gametophores, after the reproductive structure of mosses. These gametophores form from the outside in on certain branches of ramophytes and their descendants. Inside of these gametophores, meiosis takes place, creating gametes. At maturity, the gametophores will pop, releasing the gametes into the air. On the leaf-bearing branches of these plants are small pores. The insides of these pores are covered in ciliated cells that slowly drag in any gametes that land on the branch. Inside of the pore is the female gamete. When the gametes meet, they form a zygote. After the zygote matures into an embryo, it becomes encased in a sort of seed that protrudes from the pore. Mature seeds are then dropped from the main plant for dispersal. The seeds will then go on to make a new generation. Another important clarification for life on land involves color vision. As a primate, you have the ability to see 3 primary colors. Most other mammals are dichromic, meaning they can perceive 2 colors of light, usually a long and a short wavelength. Our ability to see the color red is advantageous to us because our primate ancestors were able to find fruits easier with the ability to see red. On Primin B, the tubostomes and their descendants are dichromic, detecting red and blue light. This can become a problem for some of its land descendants, as with only those colors, green, yellow, orange, and even gray can't be properly perceived. This could make it hard to differentiate between potential food and predators. The latipods and their descendants need to know the difference between these to ensure their success on land, especially smaller clades like the scanderpods. They developed another type of color receptor in their eye that can detect yellow light. This makes it harder for any potential predators to blend in. There's also some fun genetics that cause this on Earth. The cone cells in our eyes can detect colors because of opsin proteins. Dichromic organisms have 2 types of opsins, allowing them to see 2 different wavelengths of light. Trichromacy probably evolved because the gene that encoded for one of these opsins was mistakenly duplicated. The duplicate gene was then able to accumulate mutations, eventually leading to a 3rd, unique opsin protein that could detect a new wavelength of light. This color vision of latipods and their descendants, along with segmented bodies and quick reproduction cycles, allow them to diversify rapidly. This is especially true for the tiny scanderpods. This clade of small latipods are going to diversify into our new bug equivalents. This can begin as certain groups of scanderpods specialize to eat different materials. One clade may be able to eat the photosynthetic leaf-like structures of plants and the newly described gametophores. This clade's mouth will form small, sharp pincers, and their bodies will become smaller and more rounded. This clade thrives anywhere with significant amounts of foliage and contains many different species with a variety of body styles. This clade is called Pholiostoma and it appeared 5.4 billion years ago. Like how Foliostomes specialize to eat the leaves into metaphors, other clades of scanderopods can specialize to eat other parts of flora. This is similar to an ecological phenomenon known as niche partitioning. According to niche partitioning, no two species can take up the same niche in the same place. If this happens, natural selection will drive one of the species extinct or force it to take up a different niche. One clade of Foliostomes may adapt to feed on the fleshy stalks of plant life. The mouthparts on this clade become more tweezer-like to pull apart plant stalks and burrow inside. This clade exhibits a communal behavior through the use of pheromones and new chemoreceptive organs on their face. They can use this communication method to find mates of the opposite sex and avoid overpopulating their burrows. This clade is called Stipulostoma and it appeared 5.399 billion years ago. Some stipulostomes undergo further niche partitioning, this time to consume the vascular fluids of plants. This clade has blade-like pincers that they can use to score cuts into plant stalks. Primin B's plants have microscopic vascular tissues full of nutrient-rich fluid. After their stalks have been cut, they begin to leak this fluid, similarly to sap. This clade of stipulostomes continues to gnaw on the scored cuts to extract these juices. To leap between large plants, this clade has a set of springy legs. This clade went on to develop a pair of wings. It did this by modifying its ancient vestigial legs. Testoruptus, an evolutionary ancestor of scanderopods, had sets of legs that push water over their gill surface. This clade could have moved a pair of these now vestigial limbs toward its back. These limbs could then have grown into broad structures that can be used for gliding. The development of new muscles could allow this clade to flap these new wings, creating powered flight. This is similar to theories suggesting how insects on Earth developed wings. This clade is called Podolitida and it appeared 5.398 billion years ago. While stipulostomes are built to burrow into the stalks of plants, one clade of them may adapt to burrow in the soil instead. This new clade has a large pair of front limbs that are good for digging in the dirt. This clade uses their chemoreception and pheromone releasing abilities to establish colonies underground. Inside of their burrows, this clade creates special chambers where they lay their eggs. Colony members specialize to do one of two tasks: take care of spawn or find food. This clade can travel longer distances than most scanderopods to find leaf material. They tear parts of these leaves off, then use pheromone trails to find their way back to the burrow. They share their food with the caretakers in the burrow and then store the excess. These burrowers are called cuneosomes for their wedge-shaped bodies, and they appeared 5.397 billion years ago. Not all clades of scanderopods find different plant tissues to feed on. Instead, one clade takes on the predatory route. This clade is capable of eating most other scanderopods. It doesn't use standard pursuit tactics though, and instead uses a stealth method to capture prey. By using a gland in their mouth, this clade produces a viscous glue, similar to that made by velvet worms. By rapidly squeezing a muscle in its mouth, it can eject this glue to trap prey. Some species of this clade may also pre-mix some digestive enzymes in this slime, allowing them to start digesting prey before they even eat it. These predatory scanderopods are called glutostomes, and they appeared 5.396 billion years ago. The last new clade that we will be talking about today belongs to an offshoot of Foliostomes. This clade opts to use a symbiotic strategy for nutrients. They live on the integument of larger organisms, and since there is such a variety of them, different species may fill out different roles. Some may partake in mutualistic symbiosis with their hosts. acting like cleaners that eat dermal excretions and other small organisms off their host's skin. Kind of like those face mites that I mentioned earlier, which are still on your face. Other species may act more parasitic, similar to lice or scabies. This clade is called Cutosoma and it appeared 5.395 billion years ago. With these 6 new clades of scanderpods, thousands of species of small organisms are now covering the land. With small fauna now plentiful, larger food webs can begin to form. This opens a larger variety of niches for large fauna to begin diversifying on land. In the next episode, we will talk about how the current land fauna diversify to cover the continent of Inanus. Thank you to everybody who contributed artwork for today's video. If you would like to meet other people in the Primin B community, or contribute art to be featured in later videos, make sure to join the Primin B Discord. I also have a Patreon now. There is only one tier at the moment that costs $1 a month, but patrons will be featured on these end screens. The links to both the Discord and the Patreon are in the description.